In our final chapter on enzymes, we're going to start by looking at mechanisms that enzymes play. And the mechanisms that we're going to see are very similar to some of the mechanisms you might remember from organic chemistry. So if you liked organic mechanisms, I'm sure you're going to love this part. The other thing we're going to look at are inhibitors. And inhibitors are important because most of our drugs function as enzyme inhibitors. So we'll take a look at the different modes of inhibition that can take place. We'll start with catalysis, and we'll look at a common theme that we see in catalytic reactions, in that the concept of covalent catalysis. And one of the hallmarks of catalysis is that the catalyst starts and ends in the same form. But that does not mean that over the course of the reaction, the catalyst is not going to somehow change. And covalent catalysis is a great example of that, because what happens is that covalent bonds are formed with the enzyme over the course of the reaction. In general, we're dealing with a multi-step reaction, and the first step results in formation of those bonds, while some sort of subsequent steps result in breaking of that bond. We will look at an example of this in quite a bit of detail when we talk about chymotrypsin, which is a serine protease. Another example is a lot of enzymes that use ATP. If this is the enzyme here and we have some amino acid X, X would be like serine or histidine are pretty common. The X attacks the phosphorus here, and we are going to displace a portion of this. If this is the ATP molecule, we're going to displace a portion of the molecule, so essentially OR is eventually going to leave, and we end up with the phosphate portion of it actually bound to the enzyme. So that would be the first step that occurs. In the subsequent part of the reaction, some other molecule will come in and attack the phosphorus and displace the phosphate from the enzyme. Acid-base effects are very important in biochemical mechanisms. And one of the things that we very commonly see is enzymes serving as a general acid or a general base. Now, if you think back to organic chemistry, a lot of times if you wanted a reaction to occur, you would add an acid or a base, even just a small amount of acid or base. Enzymes are the acid or base that you're adding to an organic reaction. The reason for that is that, remember, we have to keep our pH at about 7.4, the pH of our cells. So we can't change the pH of the cell too much without causing destruction within the cell. However, within the active site of an enzyme, we can generate localized regions where we have a very low pH or a very high pH. And that's how enzymes function as acids or bases. They generate a very localized region within the active site where there is a pH that is much different than what we see in the rest of the cell. Now, a lot of times we will see histidine playing a role as an acid or base. And if we look at histidine, remember that the pKa for histidine was right around 6. And here is the general acid form of histidine, and this would be the general base form. And because the pKa of histidine is so close to the intracellular pH, it can very easily go back and forth between the acid and base form. A very good example of histidine in an acid-base catalysis is in ester hydrolysis. If you think back to ester hydrolysis, which was the general reaction, here this is ethyl acetate, we're essentially adding water across this bond, and we're going to end up with acetic acid and ethanol from this. This can be done in two different manners. We can, for example, add hydroxide. Hydroxide would attack this carbonyl here and ultimately displace the ethanol, or we can add an acid where the acid would protonate the carbonyl oxygen, and then that would allow a molecule of water to come in and attack that carbon. Now, in either of these mechanisms, histidine could play a role in that, either by potentially protonation of that carbonyl group, or by, in a general base form, it could actually take a proton away from water and generate the hydroxide ion. Acid-base catalysis plays a very big influence on the optimum pH of enzymes. We'll talk a bit more about this later. We'll look at a couple examples. Basically, the optimum pH deals with the protonation state of active site residues. For example, whether histidine needs to be protonated or deprotonated is going to have some influence on what is the optimum pH for this enzyme. If histidine needs to be in a protonated state, the optimum pH for that enzyme might be a little bit lower than cellular pH. If histidine needs to be deprotonated, the cellular pH is probably going to be great. And when I say the optimum pH might have to be lower than cellular pH, well, for example, enzymes that are found in the lysozyme, where the pH is around 5, they're going to tend to have optimum pHs that are pretty close to what we see in the lysozyme. So for example, there we might see a lot of histidines and active sites where the histidine is in a protonated state. Now, the other thing that can happen with pH is we can see changes in tertiary and quaternary structure. And this is because amino acid residues get protonated, and as a result, they will cause changes in possibly electrostatic interactions, maybe changes in hydrogen bonds that are forming and breaking on an enzyme. So there are going to be some changes in some of those non-covalent interactions that give rise to tertiary and quaternary structure. Cofactors are commonly used in catalytic reactions. We've seen some examples of cofactors in use, and we'll talk about a few more now. If we look at, for example, some transition metals, zinc is very commonly seen in a variety of enzymes, such as carbonic anhydrase. We'll look at it in alcohol dehydrogenase, the role it plays there. And in both cases, the zinc serves as a Lewis acid. If you remember, what a Lewis acid does is it can bind to something, for example, water, form an interaction here, and that's going to withdraw electron density away, and that's going to make it more likely that an oxygen to hydrogen bond is going to be broken, and that's going to result in something that looks like this. And ultimately we get a hydrogen ion out of that. And that's the Lewis acid aspect of it. It causes some other molecule to generate an acid, in this case water. Transition metals can also be used in redox reactions. Iron is very often used in the cytochrome P450 enzymes. The iron goes back and forth between an iron in a 2+ and a 3+ oxidation state, where it will release an electron. And there are a variety of enzymes that we find mitochondria, where iron goes back and forth between this 2+ and 3+ oxidation state. We'll actually look at some of these when we talk about electron transport. Alkaline earth metals like magnesium and calcium can play roles in enzyme mechanisms. Magnesium in particular forms complexes with ATP. So very often where ATP is used as a substrate, magnesium will form an interaction, as we see here. And what it does is it simply makes this bond more easily broken, so that the transfer of that phosphate ends up being a lot easier than it normally would be. One of the things that enzymes do is they put the substrate in the right orientation that the reaction will occur. And when it does that, very often we put strain on the substrate and strain on the enzyme as well, such that the bonds in the substrate that need to be broken are broken. Also, the bonds that are going to be broken are in the right place for the formation of new bonds that are going to occur. A great experiment that looked at a very simple system and the rate enhancement that it could occur was the formation of these anhydrides that we see over on this side of this figure. And in the first reaction, we have an acetate ester reacting with acetate to form acetic anhydride. And this relied upon the two molecules coming together and coming together in the right orientation such that this oxygen here was attacking that carbon. The likelihood of that happening is somewhat low unless you have an incredibly high concentration of the substrates. Now, going down to the second reaction here, we are putting the two reactive groups that need to react very close to each other by having them bonded together by sticking those two additional carbons in there. And what we see is a rate enhancement of about 100,000 fold. And the key element here, though, is that all of these bonds, this bond, this bond, and this bond, can rotate. So although everything is relatively close to each other and the two groups that are going to react with each other don't have to diffuse together in solution, they're already there, there is still possibility that they're not going to have the right orientation to undergo that reaction. So if we take this a step further and make it so that we can't have rotation about these bonds, so this bond here we can rotate, this bond here we can rotate, but this bond right there is locked in place. So as a result, this carbon and this carbon are always going to be in the same place, spatially speaking. So as a result, we just have to rotate about this bond here such that this oxygen can then attack that carbon. And from that, we get another 1,000-fold enhancement in the rate. And this is an illustration of what we call proximity effects, meaning that the substrate has to be in the right place and the right orientation to react. And it has to react both with the catalytic residues as well as with the other, if there's another substrate for the reaction, it has to be able to react with that. So, and this experiment here, although it's not an enzyme catalyzed reaction, it shows the power of enzymes in terms of the ability to bring together the substrates in the right place as well as putting them in the right orientation in order to make them react. The further effect that an enzyme has is in what we call strain effects. And this is what allows for the formation of the transition state. And it goes back to stabilization of the transition state. So the transition state is a very strained species relative to the substrate. And if we can put strain on the substrate such that it's going to be more likely to form the transition state and then stabilize that transition state, that's going to help the reaction occur. And that's one of the effects of what we see in an enzyme catalyzed reaction, is that the enzyme tends to stabilize that transition state, thus making it more likely that the reaction is going to proceed. Temperature and pH dependence are two unique things to enzymes, and I'll talk first about temperature. I put here that most enzymes have optimum temperature at organisms' body temperature. Notice I put body in quotes because while we as humans have a body, bacteria do not. Bacteria are single cellular organisms, so they are subject to the temperature of their environment, which can change drastically. Additionally, if we think about cold-blooded organisms, such as reptiles and fish, their body temperature is going to change drastically depending upon their environment as well. So going back and being a human and thinking about humans, because very often we are going to focus on human biochemistry in this course. We operate at 37 degrees Celsius. That's 98.6 Fahrenheit for the non-advanced world. 37 degrees Celsius is the optimum temperature for most of our enzymes, or somewhere relatively close to 37 degrees Celsius, probably within about 5 degrees Celsius. Now, one of the things that we say is, well, let's go ahead and throw on this plot here. Let's put 37 degrees Celsius as that point right there. Below that temperature, we see a slow decline, and most of this decline is simply due to, in an aqueous solution, as things get colder, they tend to, reactions don't move as well, simply because the molecules aren't moving as quickly through water. So a lot of the decrease here is simply due to decrease in the diffusion rate. Whereas if we go higher than this, all of a sudden we notice this very steep cliff. Right here, what's happening is we're seeing denaturation of enzymes. And with our enzymes, a lot of our enzymes tend to start denaturing above about 42 degrees Celsius. This is why a prolonged fever is not good for people. Once you have a prolonged fever for a long period of time, you start to see denaturation of proteins, and with that, denaturation of enzymes. This is why we want to keep our body temperature relatively constant at about 37 degrees Celsius. Looking at pH dependence, as I mentioned before when we talked about acid-base effects, enzymes need to have amino acids in the proper protonated state for reactivity, for the reaction to occur. So if we look at two examples of, these are both proteolytic enzymes that hydrolyze peptide bonds. Chymotrypsin that we see over here has an aspartic acid residue and a histidine, and the aspartic acid residue has to be in a deprotonated state, and the histidine, what happens to it is that over the course of the reaction, the histidine goes through a protonated and deprotonated state. So it has its pH optimum up here at about 8. Normally it functions around 7.4. It actually functions in our small intestine, where it breaks down the proteins that we eat in our food. Pepsin, on the other hand, is active in our stomach, and it has a pH optimum right around 2. And what we see in pepsin is that there is one aspartic acid that is in a protonated state and another that is in a deprotonated state. The pKa for aspartic acid is around 4. So having and this is one of those cases where the pKa of the aspartic acid actually slightly changes in the environment of the enzyme, and it is adjusted down to about 2 because that is the approximate pH of the active site. And at that point, we see one protonated aspartic acid, the other deprotonated, and that's what actually allows the reaction to occur. So a work problem for you guys to do. So the first one is transfer phosphates from one molecule, such as ATP, to another. Often involves a phosphohistidine intermediate, the phosphate bonded to a histidine residue in the active site. Draw the structure of such an intermediate. So I'm going to take ATP, and I'm going to actually refer to it as ADP with a phosphate on it. And I'm going to take a histidine residue, and we're going to have nitrogen, and I'm going to put my extra lone pair of electrons on this side. Sorry, meant to put the double bond there. And what's going to happen is we're going to do an attack from the nitrogen to the phosphorus, and then we're going to be breaking that bond. So that's going to go off as ADP, and we're going to end up with our histidine residue. Bonded to phosphorus here. And our nitrogen is actually going to have a positive charge on it. That is our phosphohistidine intermediate. Next problem, if an enzyme uses a deprotonated cysteine residue in the active site, what structure would the cysteine have? So normally, cysteine has a structure that looks like this. We're going to deprotonate that so that it looks like this. And sorry, that's a negative charge. Would you expect the enzyme to be more active at pH 6 or pH 8? Well, the pK for this is about 8.3. So pH 6, we're going to expect to have more of the protonated form. Whereas at pH 8, we're very close to the pKa, so we're going to expect to have a pretty good mix of these two forms. So I would go with more active at pH 8.